our channel, the Pandora Pharma. Today's our new video is on identification and authentication of herbal materials. We will look in detail of the study of herbal materials. So the introduction part, we are seeing that the herbal materials are plant-derived substances including leaves, roots, stems, flowers, seeds, or their extracts used for their therapeutic or health promoting properties. There is need for correct identification and authentication of herbs. Okay, why? To ensure the correct plant usage. Ensure correct plant usage means that potential misidentification can lead to the use of the wrong species or plant parts. Some herbal drugs have toxic look like Atropa belladonna versus Withania somnifera. Accurate identification ensures therapeutic consistency and avoid adverse effects. Next, the support standardization and quality control. How it happens is that authentication is the first step in standardization of herbal materials. It ensures that bioactive constituents are present in the expected quantity and quality. It prevents the adulteration with standard or inert material. This is the introductory part. We will look further more in detail. So, it meets regulatory and pharmacopoeial requirements like what? Regulatory bodies, example Ayush, WHO, US FDA, mandate botanical authentication like pharmacopoeias specify microscopic and microscopic and phytochemical parameters for identity. Ensures compliances with good agriculture and collection practices and good manufacturing practices also. Protects consumer safety, which prevents toxic reaction due to substitution or contamination, which reduces the risk of drug herb interaction by ensuring correct identity. It builds consumer trust in herbal products. So basically, the identification and authentication is very important for the identity of a herbal drug, as it is going to go for patient or human use. So it should be safe, non-toxic, and of real quality product. Next is that how herbal drug identification happens. So it is the process of determining the true botanical identity of plant material or its part used in a formulation, ensuring it matches the correct species, genus, and plant part as stated in pharmacopoeias or references. So what are the objectives? To confirm the correct source, botanical origin. Okay. To ensure consistency and quality of raw material, which is the material. To provide a scientific basis for standardization for herbal drugs. These are the three that we will go for. Next, what are the challenges of adulteration and misidentification? So, the herbal medicine industry faces significant changes in maintaining product integrity due to morphological similarities between the species. Even experienced that the botanist and pharmacognosist struggle with identification with plant share similar physical characteristics. This happens that they faces the problem of identifying which is the real source. Okay, so dried, powdered, or processed material present additional challenges as diagnostic features may be obscured or destroyed during preparation. This is the factor. So, a global survey using DNA based method found that about 27% of commercial herbal products were adulterated. The content did not match level. In the study of raw material or the raw herbal trade in South India, around 12% of market samples were found to be adulterated. So, what are the methods or the main methods you are going to see in the identification? They are they can be organoleptic, macroscopic, microscopic, physiochemical, chromatographic, spectroscopic, and molecular. So, in organoleptic, so organoleptics are like using your organs, you're identifying the quality of a material. It's like the sight, smell, taste, touch, and something hearing to identify and access the quality of a material. Okay. It feels now, for example, like your grandma says that this type of sound you will get in market, that one product you have to buy. Or sometime it's like this type of green color leaf you have to buy. Okay, so the size of this fruit should be like this much, the shape should be like this much. Means the quality, the identification happens uh, by the senses. Okay, so for example, for the medicinal plants, it's like recognizing dried ginger rhizomes by its bridge color and wrinkled surfaces. Okay, next, that identifying that peppermint leaves by their characteristic menthol smell, distinguishing neem leaves by their bitter taste. Okay, licorice roots, fibrous texture, feeling it, cracking sound of dried bark like cinnamon. Okay, next we will see that how macroscopic things happen. So, macro means large. So, visual examination of whole or cut plant parts, leaves, roots, stems, flowers, fruits, to study external morphological features. It's like the ginger rhizome, dried ginger rhizome, okay, which we call sort. So, its size and shape should be 2 5 centimeter in length, 1 to 2 centimeter in thickness. The tulsi leaves, it should be ovulate with serrated margins. Okay, like the surface characteristic of cinnamon bark, like a rough woody surface with longitudinal furrows, the fracture texture of licorice root, fibrous fracture, cardamom, the brittle fractures, hairiness, presence of texture, exudates, marking, veins, aloe vera leaves, thick, fleshy leaves with clear latex. These are some features that we are going to see for the leaf, root, stem, flowers, fruits. Now, the qualitative microscopy. So we can see that the stomatal numbers, stomatal index. Okay. तो चलो बच्चों, अब थोड़ा सा अपनी वेव को राइट मोड पे लाते हैं और स्पीड अप करते हैं अपने इस लेक्चर को। दैट द स्टोमेटा नंबर क्या होता है, तो एवरेज नंबर होता है स्टोमेटा का पर ऑफ एपिडर्मिस। एपिडर्मिस समझते हो? एपिडर्मिस लीफ का पार्ट होता है जो कि ग्रीन पार्ट को हम देख रहे हैं। तो ये क्या हेल्प करेगा कि स्टोमेटल डिफरेंसी डेंसिटीज़ को बताएगा। ये डेंसिटी मतलब एक पर्टिकुलर पार्ट में कितना ज़्यादा डेंस अमाउंट में स्टोमेटा प्रेजेंट है। ओके, सो जैसे कि देख रहे हैं कि डिजिटलिस पुरप्यूरिया इस स्पीसीज़ में 120 से 150 स्क्वायर में होता है, वाइल डिजिटलिस लनाटा में 180 से 220 स्क्वायर में होता है। तो ये हमको आइडेंटिफाई करने में बहुत ईजी पड़ेगा। नेक्स्ट, स्टोमेटल इंडेक्स, द रेशियो ऑफ स्टोमेटा टू टोटल एपिडर्मल सेल्स। अब ये एक रेशियो है, ये इंडेक्स, ठीक है। इससे क्या वेरीफाई करेंगे हम? कंडीशंस को, स्पीसीज की स्पेसिफिक कंडीशंस को एग्जांपल के थ्रू देखते हैं। दैट द धतूरा मेटल is 18 to 20%, while Dhatura stenomium is 22 to 25%. So one plant, different species of different location, but we can identify it very easily through these indexes. ठीक है, तो ये क्वांटिटेटिव माइक्रोस्कोपी है। Again, the second thing that we can see that is the stomata type. So look at the diagram. This can be dicytic, anisocytic, anomocytic, parasitic, actinocytic. So this is based on that how many number of epidermis are covering or giving surface to the middle stomata. ठीक है। Stomata are multiple opening found in epidermis of the leaves or young stems. It is a key taxonomical character for the species. It is a key taxonomical character and a constant for a species. Okay, for example, Dhatura stronema shows anisocytic stomata. अब ये एनिसोसाइटिक स्टोमेटा ही शो करेगा और कुछ शो नहीं करेगा अगर हम इसको आइडेंटिफाई करते हैं। तो वाई द नरियम इंडिकम शोज पैरासाइटिक स्टोमेटा। नेक्स्ट इज ट्राइकोम टाइप्स। These are the types you can see in the diagram, that this is the cinna which is having trichomes, okay, like unicellular watery trichomes, labelia, cannabis, and vasaka, digitalis. These are separated tube-like rectangular, okay, so they are elongated outgrowth of one or more epidermis cell. एक और एक से ज़्यादा एपिडर्मिस सेल के एलोंगेटेड पार्ट्स होते हैं, ठीक है, समझ गए। ये क्या हेल्प करते हैं आइडेंटिफाई करने में और डिटेक्शन में ऑडल्ट्रेशन के लिए। हमारे पास कोई ड्रग आई, हम उसका ट्राइकोम टाइप देखेंगे और चेक कर लेंगे कि वो ऑडल्ट्रेटेड है कि रियल। सो कैरेक्टरिस्टिक ऑफ मैनी स्पीसीज कुड बी सीन। एग्जाम्पल इज लाइक डिजिटलिस purpurea कवरिंग एंड ग्लैंडुलर ट्राइकोम। ग्लैंडुलर ट्राइकोम होता है डिजिटलिस purpurea में, while cinna has unicellular waxy trichomes. This is very important, note it up, okay. Next we'll see that starch grains. So starch grains are like potato starch grains, maize starch grains, rice starch grains. The size is different, the density is different, okay. So they are elongated outgrowth of one or more epidermal cells, same as trichomes. It helps in identification or detection of adulteration, same as that. So the covering. Next, chromatographic and spectroscopic identification and authentication. So chromatographic techniques revolutionized the herbal authentication by enabling the creation of unique chemical fingerprint. हर प्लांट का, हर ड्रग का जो एक्टिव कंस्टिट्यूएंट है वो एक फिंगरप्रिंट रखता है, ठीक है, फॉर प्लांट स्पीसीज। ये क्रोमेटोग्राफी और स्पेक्ट्रोस्कोपी की टेक्निक से ही पॉसिबल हुआ है, और ये बहुत बड़ा रिवोल्यूशन है आइडेंटिफिकेशन और ऑथेंटिकेशन की फील्ड में, ठीक है। नेक्स्ट इज दैट डिटेक्ट एंड क्वांटिफाई द कॉम्प्लेक्स मिक्सचर ऑफ सेकेंडरी मेटाबोलाइट्स, okay, that are characteristic of botanical material. How these systems are present, like thin layer chromatography, TLC, high performing thin layer chromatography, HPTLC, high performing liquid chromatography is HPLC, gas chromatography, GC, column chromatography, paper chromatography, LCMS liquid chromatography mass spectroscopy, GCMS gas chromatography mass spectroscopy. So these are some things which we'll learn in detail, or we'll see that these could be seen for the identification. So like the TLC, you can see this plate. Okay, so this is a silica covering over aluminium plate, and markers or samples are spotted or banded on the one side, and uh, for uh, for some polarity of separation based and affinity to stationary phase. It is used to identify and fingerprint profiling of herbal drugs. So like profile of Phyllanthus neurori can be observed from 15 different locations of India. Fingerprint region for compound remain the same. हमने एक प्लांट स्पीसीज जिसका नाम है फाइलेंथस नायरूरी, उसके 15 डिफरेंट लोकेशन इंडिया से उठाए, पर उसका जो फिंगरप्रिंट रीजन है उस कंपाउंड का वो सेम है, ठीक है। तो इस तरीके से हम HPTLC में, इसमें हम ऑब्जर्व करते हैं सेम प्रिंसिपल से, बस होता क्या है, ये एडवांस है। माइग्रेशन ऑफ कंपाउंड बिटवीन स्टेशनरी फेज एंड मोबाइल फेज, बट द ग्रेटर प्रिसिशन, रिजोल्यूशन एंड रिप्रोडक्टिविटी। सो एप्लीकेशन टू कंट्रोल सॉल्वेंट फ्लो, जेंटरिफिकेशन, स्कैनिंग, एंड इसमें लैम्डा मैक्स निकाल सकते हैं हम उसका, और कैसे पीक्स हैं उसको कंपेयर कर सकते हैं काफ़ी ईजीली, ओके, स्कैनर से, कैमेरा के थ्रू। HPLC is also there. Liquid chromatography, mass spectroscopy is also there. It identifies the non-volatile thermomobile phytochemicals, okay, for the sheet cell and detection of 5 peaks within the detection time, like these are the time, the fragments and the results. We'll look further more videos for the detailed version of these studies, okay. So this is the conclusion, that the identification and authentication of herbal materials from foundation of quality assurance in herbal medicines through the integration of classical techniques, macroscopic, microscopic and physiochemical, with advanced chromatographic and molecular tools. We need that. The purity, safety and efficacy of herbal formulation is so important for the herbal industries. Ending up that authenticity is the first step towards safety, and science is the bridge that connects tradition to truth or the trust. So thank you so much, and again welcome to Pandora Pharma. This is a platform for learning, and new magical videos will come to you for your study purpose. This is an higher study topic from the pharmacognosy part of your M pharmacy study or B pharmacy study or botany study, so you can use there so easily, okay. Thank you. Like, subscribe and share.